That's the main one.
So you
it looks like it's the full screen, so you're fine.
It's a full screen. Okay, perfect. So I have a bigger screen because my eyes have aged a little bit. So I'm not kind of ignoring you. I just look at the other screen. Okay, if you look at me. Okay. Well, thanks for the invite. And as you have already realized, is like I don't have an American accent, and it was already mentioned. I did not grow up in the US. I grew up here, which is in Switzerland. So that's the house, and I was actually born at home in the room up there under the roof. For me, Switzerland was not, I don't know, was a difficult place because I hate winter. So every spring, what you have in Switzerland is that, like, you know, the snow melts away and then the trees get in flower. And you can see here that my grandfather who built that house, 1901, he actually kind of really liked his garden. So we had all these kind of fruit trees around, and the house still there, and the fruit trees are still there as well. But every spring, you know, when this dreadful winter and the darkness was over, I went in front of this house and I could hear the bees buzzing, and I could even smell them. And a few weeks later, we had bumblebee colonies in the garden, which was absolutely fascinating. So for me, like, I had a very early exposure to bees. I call it the golden age of bees because bees were all over the place, and they were healthy, and we had them in our garden. And for me, they signaled spring, right? So a time where, like, better times basically approached again. Now, as you all probably know, that golden age for bees eventually came to an end. We have heard about, like, declines of bees. I think it started in honeybees because the beekeepers that keep them realized that first that they lost all the hives. I realized that when I did my PhD in Zurich because we had more and more problems to catch bumblebees, right, for our research. But by now, I think, like, we have even gotten a step further, right? So this is like an article that appeared at some point in The Guardian that basically now rings the alarm bells that we have something like what they refer to as insect Armageddon or insectageddon, right? Where we see massive declines in insects. And it's really important that we all keep in mind, I don't have to remind you about this, but the vast majority of these insects that decline or that we lose are our friends. They're not our enemies. Right? We basically have increasingly more problems to keep our friends alive and to deal with the really nasty insects, which we almost have or we lose our kind of ability to effectively regulate them. And then, of course, that kind of decline is especially dire when we look at pollinators, because first of all, they're really important for what I would call ecosystem stability, but they're also really important because they produce our food, right? More than a third of what you eat depends on insect pollination. Bees are the main pollinators. It's about 80 crops of agricultural interest that has a value of, I think, $220 billion per year. So it's an important part. I think as of today, we actually have a really fairly good idea why we lose at least the pollinators. First of all, we know that bees harbor a lot of parasites and pathogens, and they can, some of them, the bees can actually deal with really nicely, but others spread globally and can result in major collapses of bee populations. But it's also like environmental change, and I think that has two different kind of aspects. First of all, it's climate change, which disrupts the life cycle of the plants and the flowering with the life cycle of bees, especially with solitary bees or bumblebees. But it's also that, like, we have lost so much areas for foraging habitat that the bees need, right, for a healthy life. And then we have, of course, with the increased demand in food, with the industrialization of food, with a green revolution, we need to protect our crops. And that means that we have to use increasingly more aggressive pesticides and insecticides to protect these crops. And the pollinators don't like that. We know that there's ample of evidence that these pesticides have sublethal and even lethal kind of effects on pollinators. And finally, it's also like, you know, the beekeeping itself that has changed so much over the last decades and centuries. The way we keep bees now is very, very different from the traditional way how we would keep bees. And that has certainly kind of impacted the health of bees. Bees are just not made to be traveled around for 14,000 miles on a highway. Well, we know that. So I think really early onwards, during my studies in Switzerland, I got really worried about bees, and very early onwards, I wanted to save the bees, or I wanted to do something to support like the survival of the bees, like this bumblebee here, which is sitting on my finger, a queen in the spring. So I applied for a PhD position at the Swiss Federal Institute of Technology, and was invited for an interview, and my supervisor worked on bumblebees, and I said, well, I want to do something in the area of bee conservation. And he said, that's fine. And he proposed me the following project. He said, like, what I need is, we need to understand like how the genetic diversity that occurs inside hives or among bees actually contributes towards the protection against diseases. Now, initially, I have to be honest with you, that was not exactly what I was looking into, but you know, it's like as a PhD, we thought, well, maybe we should look into this, because there was like there were papers published at that point that basically said, well, maybe we need to really understand how genetic diversity inside and outside hives basically contribute to the health of bees. And so I started with bumblebees, and my initial idea was to basically manipulate genetic diversity inside hives, and then kind of see how that would kind of protect the bees, for example, if you have more genetic diversity. And I show you this picture because you can see a mating bumblebee pair here. And the idea in the beginning was to have queens that are singly or multiple mated, and then compare their performance. And when I did that, I hit a little bit of a brick there, because I could easily get a first mating. But when I wanted to have a multiple mated bumblebee queen, and I added a second male to the queen or to queen's cage, the queens become really hostile, and they started to even sting my males to death. So in the end, I had like 3-4 dead males at the bottom of my cage, which was like a little bit frustrating. So we decided to take over management then, and to develop an artificial insemination technique for bumblebees, which eventually worked. So you can see this here as in a schematic way. You have a glass capillary, and that glass capillary is introduced into the queen's sexual tract. You release the semen that you have collected before, and there's this like little organ here that we call the spermatheca, and that's where the queen then stores the sperm for an entire lifetime. So with that technique, I was able to actually manipulate genetic diversity in bumblebee hives. And I then had a look like how much parasites do they actually have. And when you do this, it's very clear that like if the queen mates multiply, the colony has less parasites and they produce more offspring. So they have more young queens and they produce more males. So it's a tremendous advantage. And a little bit later on, it was shown that we have exactly the same effect in honeybees as well. So that actually opened a can of worms and more questions that were answered, right? Because what I said before, the queens don't take advantage of that. Queens mate only a single times in bumblebees. It's a marriage for life, and they will never remate again. And the question is, why would they do that if they get such a big advantage out of multiple mating? And so like we started to become increasingly more interested in the mating biology of these these bumblebees. And so what you do is like in this case, like we let these bumblebees mate, and we then throw them into liquid nitrogen so we can stop the mating process at different times, right? And we can look at what's happening inside. So after a short while, when they mate, what you can see is that the male ejaculate appears inside the queen's sexual tract. And that's a bee ejaculate that was dissected out of a bumblebee queen. And you can see that there are two parts. First of all, this white stuff at the top, that's the sperm. And then we have this huge gray mass that we refer to as seminal fluid. And when I saw that, the first time I was really puzzled about this, because it's clear that the queen needs a lot of sperm to fulfill like her duty as the queen and build that entire colony. But why is there so much seminal fluid? So what we did is like we collected the seminal fluid and we used this artificial insemination technique that I showed you before, and we injected just the seminal fluid into queens. And what we found is that the seminal fluid acts as a chastity belt. So if the queen receives that substance, she stops mating and she becomes hostile against other males, which is remarkable, right? So obviously the male has power over the queen. It's the male that basically determines how often the bumblebee queen is mated. But how can a liquid do this? So I started to kind of become very obsessed with like what is the seminal fluid and how can it actually do this type of things? Now, this is like outside of the realm of like the type of work that I was doing at that point, which is like more on the ecology and the evolutionary side, right? If you start to think about molecules or setups of things of interest, like of seminal fluid here, you need like a different kind of discipline in biology, and that's biochemistry. And at that point, there was like a very famous center that was able to actually kind of identify what is in such kind of substances or in such kind of tissues, right? And that was in Western Australia. So I had to move to Australia in order to be able to kind of do this type of analysis and have the expertise around to help me with that. Now, Australia doesn't have bumblebees, so I had to shift to another bee species as well. And so I started to work with honeybees. And apart from the fact that they occur and they are really healthy in Australia, there's also another kind of advantage of it. Honeybees have a lot of parasites. They're actually kind of that social insect where we know the largest number of parasites that can harbor in these beehives, which is quite remarkable. On average, a social insect, an ant, a bee, or a wasp has about, on average 1.8 parasites. For honeybees, we know more than 40 and maybe even up to 80 different parasites. So it's really kind of dramatic how much more diseases honeybees have. But there are also kind of interesting kind of observations about honeybees. The first one is like the queens in these species actually mate multiply, so they mate with several males. And the queen is able then to store fractions of that, only a small fraction, about 3% of the sperm that she collects during mating. She stores in that spermatheca and she can use that for up to 7 years. And imagine that, that is quite remarkable that an individual can store sperm for such a long time and can use it so economically, right? So there's a lot of kind of secrets that like wait there to be discovered. So what we can do then, if you're interested in seminal fluid, is we can breed males and we can dissect them. And these are the sexual organs, the internal sexual organs of a male honeybee. And it consists of two parts. It consists of seminal fluid, which is stored in these accessory glands here. I have to show this here. And then we have like these pink parts. That is where the mature sperm is stored. So I was interested in that seminal fluid. Right? So I collected that from these glands. And I used then a technique that I was exposed to the first time at primary school. And I don't know whether you have done that, where you have like this blotting paper and you put a marker pen dot on it and you put it in a glass of water, and then the water basically rises in there. And as it does, it separates the different colors. Has anyone done that? Or is that like in the iPhone world? And so it's like, yeah, you have done it as well. Okay, excellent. So we use a almost identical kind of technique in the beginning, where we basically collect the seminal fluid and we separate that. And in this case, this is what the end product looks like. It's not only separated in one dimension, like my blotting paper, it's separated in two dimensions, right? So you can see these blue dots, and each of these dots then represents what I refer to as an agent, right? So these are individual molecules that are in there, and you can already see there are quite a lot of dots, and then there is like some bigger dots, so there's more of that around, and we have very small dots, right? So in the future, I will refer to as my little secret agents, because in the liquid you can't see them, but underneath they are active and they're doing things, right? The big advantage and the reason why I moved to Australia is that they had the necessary infrastructure, in this case, an entire floor full of mass spectrometry equipment, and those machines can identify what these dots are. So we get names, we get genes, we get an idea of what these substances actually are. And if, like, I'm not going through this list or kind of looking at individual proteins, but what we've generally found is that, like, we have three main groups of proteins that are in seminal fluid. The first group keeps sperm alive. Well, that's something that, you know, you probably expect from seminal fluid, right? And then there were, like, these agents that seem to be involved in some forms of warfare. So they are normally kind of doing nasty things. And if you think back about bumblebees, that was an expectation that we could have had as well, right? So some modifications, some conflicts between males or between males and the queen over the mating frequency. And we also found immune kind of genes in there. So the seminal fluid in bees is like a Swiss Army knife, right? So it is like it has multiple functions, and this technique basically allowed me to identify individual agents, and I started to kind of study them in further detail. And of course, I was really drawn to these warfare agents, right? So the way we kind of approach that is that we bred again males, right? Here, so two kind of honeybee males, and we can dissect their sexual tracts, and we can then basically collect seminal fluid and the sperm, but we do that separately, right? And what we do then is, like, we set this up where we use the sperm of what I call a focal male here, so that's the one on the left, right? And we then present that sperm to the seminal fluid of either the same male or of a potential rival male that could mate with that queen as well, right? And we leave that sitting on the bench, and then after about 20 minutes, we ask how happy that sperm still is, right? And we have a way to measure sperm happiness with two fluorescence dyes. And the green ones here on this picture here, these are the happy sperm for the reason because they are alive, right? And then we have unhappy sperm. They're red because they're dead, right? So we can assess, like, sperm survival in these fluids. And what we find is that if sperm comes in contact with the seminal fluid of rival males, they kill it. So males are able to use the seminal fluid to kill the sperm of rival males. Now, this in the long term cannot be in the interest of queens, because the honeybee queens also only mate at the beginning of their lives. They store all that sperm in their spermatheca, right? So if this goes on and on and on, the queen will soon run out of sperm. So our prediction was that the queen eventually will have a say as well. And that's exactly what we found. The queen, after a while, will intervene into this conflict between the males, and she can stop it. And we know the substances that are involved in that. If you got a bit confused of, you know, about the mating biology of honeybees, here's like a picture that basically nicely summarizes what's going on in honeybees. So we have an arena. The queen sexual tract is an arena. The males enter that arena as ejaculates. The seminal fluid it acts as weaponry, which they use to kill each other. And then you generate, like, complete losers, like that guy there in the back. You generate winners. And then you also have, like, others that are left at the mercy of the female. I assume you know that tradition in the old Roman kind of arenas, right? Like where the females up there, they have the thumbs up or the thumbs down. So in the end, like, what we have here is, like, that the males fight for paternity, but the females actually rule in honeybees. And so that was one part, but, like, it was not the end of the entire story, because if you looked at these agents, there was more than just that, because there were also agents that were highly specific in targeting neuronal kind of tissue of bees. So what we did is, like, we artificially inseminated seminal fluid into honeybees, and then we studied the brain, right? So this is a cross section through one of our queens. And you can see here in the middle part here, you can see this brain, right? And based on these kind of cross sections, we could actually get this out of the queens, and we started to see how active that brain is, how active the genes or the gene expressions are in these parts. And what we found is that if the queen is exposed to that seminal fluid, the activity of the brain changes. And when you look at like what is targeted, it is those genes, it's those molecules that are responsible for the eyesight of the queen. Right? And there are some interesting ways how you can test that. So here's like a way we tested that. So we have here a bee. This is a worker bee, but we did it with queens as well, which is kind of held in place here. And then in the back here, we have this little diode, and this diode is basically flickering light. And we are able to basically measure how accurate the queen is able to detect that light, whether she's able to detect it at all, or whether she's detecting to detect the flickering. And if you do that, what you see is that if those queens that receive seminal fluid, they have a reduced ability to see. So with other words, like what we came up with is here the idea that the males are able to blind the queens. Now to test this in the field, we again inseminated queens with seminal fluid, and we glued these little radio frequency tags on queens and let them fly to see whether, you know, after the mating flies, when they leave the hive and afterwards come back, whether how often they go out and whether they would actually find back. And what we found is that like queens that have been mated and still go out, they have a higher chance, a significantly higher chance that they get lost, and they also make fewer mating flights. So it seems that the males actually are able to fight with other males over paternity, but they still have also the ability, like in bumblebees, to manipulate the queen and to manipulate her mating frequency. So this is about this part about the warfare. We also found immune defense agents in our seminal fluid. And maybe that was not too surprising, but what was surprising is the following. So this is the immune system of the honeybee. And okay, I know it's really complicated, but that's already some statement I want to make here. Like the bee immune system is nothing simple, even if you might have heard that before. Right? It's very complicated. I think we have not even reached slightly the bottom of how complicated it is. But what I have here is like at the top here, what you have is like baddies that come along, like fungi, bacteria, injury through mites and stuff like that. You have then a recognition system here, which has odd names. Don't ask me why they're given these names. And then we have a response to this. Right? And so if you look at like, what of these do we actually find in seminal fluid? I can put red stars on those kind of components that we find that you can see here now. And that really kind of was, wow. Right? So what you can see here is that we detected almost the entire immune system to be present in seminal fluid. And the first thing is like, for me, that is like super exciting because now I can study the immune system in a liquid in a vial on my bench. Right? And the second thing is, of course, why would they do this? Because this is costly to produce for the queen. This is not something you just do. If you upregulate your immune system, you sit in your bed and you have a fever and you're taken out of action for 3 weeks, right? or 3 days. So why would they do that? And I punished one of our PhD students to find out why. And it took her a while until she came to my office with this very picture here and said, I got it. Right? So she showed me this picture here, and you can see here an ejaculate of a honeybee male. So you can see the sperm here, but she also found these white dots here. Can you see them? And that's a fungal disease. It's called nosema. So what we found is that bees have sexually transmitted diseases, which was something new as well. Right? So sex is not without risks in honeybees. Now, the reason why we went there is that I found these immune agents in seminal fluid. So what happens if we collect seminal fluid and we bring that together on the bench with these spores? And this is shown here. You have here like the survival of these nosema spores. So how much is still alive after about 20 minutes? And you have a control. So that's just like we dilute them in some water. And then we have water with seminal fluid one. And so you can see that like in the control, almost 100% of these spores are still alive. If you have the seminal fluid around, that kills almost 90% of the spores straight away, puff. right? I was really like, wow, you know, that was the first time for me that I saw honeybees taking on a real parasite and actually neutralizing that. So I think that that was really kind of encouraging. But how do they do this? Right? So we looked a bit more closely into this, and here you see a lot of dead parasites, and I like to really watch dead parasites, especially after the last 3 years, and you probably as well. So dead parasite, the interesting thing is they are dead for 2 reasons. So on the one side here, we just have dead spores, and that is done by a subfraction of the seminal fluid, which contains only very small molecules. And then we have like another fraction of the seminal fluid, which has larger agents, larger molecules in there, and they kill them slightly different. You can see like these, that they are empty, and there are these little strings looking out. Right? So what the bees do is they either kill the spores straight away, or they have agents, and the agents actually trick the parasite. The parasite is in the gut of these bees, and the parasite looks for gut cells. And then once it finds a gut cell, it basically extrudes this little line here, and it penetrates the gut cell of the bee and delivers the DNA in there. Now, the agents in seminal fluid actually give these spores a wrong signal. They tell the spore that it is in the midgut, and they immediately kind of shoot out these little kind of strings, but there is no cell. Right? Its seminal fluid doesn't contain any gut cells. So the spores actually shoot their DNA into no man's land. It gets digested away, which is pretty nasty way to kill a parasite. But what we found in the end is like we have redundancy and we have specificity. Right, so the bee immune system being much more complicated than we might have considered in the beginning. But does it actually work? Right, so we know the agents, we have identified the agents. So what we did is like we started to collect bees, and we fed them with the disease straight away, and we then keep them in these little boxes for up to 3 weeks. But after 6 days, we started to provide them with these agents because we can buy them. We can buy them from chemical companies, right? So we added these bee immune kind of agents that came from a company, and we fed the bees with them over about 3 weeks. And we were interested in like, does it have an effect, right? And so what you can see here is like we have these 2 controls. The positive control is those who got spores, and the negative controls is that did not receive spores. And both of them got infections, right? You see they got quite a number of spores in their gut. So what happens with the 3 agents that we had there? And that's what is happening. So each of these 3 substances by themselves decrease the infection rates tremendously. Right, so we suppress diseases in bees. Now we can also look at it in a different way. You can say like, well, okay, but it's still there. So what you can also do is you can, instead of looking at how many diseases are there, you can look at these bees and say who got it or who lost it. Right. So this is here. We have the 2 control treatments, and you can see that's the percentages here on the one side. After about 3 weeks, 60% of these bees still carried the infection. So how many of the bees carried an infection that were treated with these agents? And that's significantly less, right? Only about between 15 and 20%. So in other words, what you can see here is we are not only suppressing the infection, we're even curing some of these bees. They completely lose the infection. And so that's really cool because bees have active immune systems. They can defend themselves, and we can see that they can do this. Right? Now, if you do this type of research, it doesn't take long until, you know, people become interested in it. And in this case, of course, the beekeepers, which are the guardians of these animals, and they struggle to keep their bees alive. Right? The US loses about 40% of their bees each year. So, not surprisingly, bees knocked on our doors and asked whether we can help them, whether we can collaborate on this type of research that we had done. Right? And that's what we started. That is going on now for more than 18 years. I think we had the tradition, or we introduced the tradition that we organized some bee health workshops, where we basically bring the beekeepers to the campus and we discuss like what is really worrying them, or whether the type of things that we develop would actually help them to keep their bees healthy and alive. And I just mentioned that the next one is coming up, hopefully, in Riverside between the 9th and the 11th of September, where we have one of these meetings again. Now, if you kind of talk to the beekeepers, especially here in California, we realized that like Southern California, the southern part here, is absolutely unique globally. And it has a couple of reasons why that is. And that was one of the reasons why I actually moved from Australia to here. I think this is an absolute dream to work on bees in the southern part of California. And the reason for that, first of all, is with UCR, we have a campus here which has a very long tradition doing entomological research. If you look at the world ranking, and that's not my doing, right? This is the people that have been here for decades. The research that has been done here at Riverside in the entomology department is extremely highly ranked. The department is ranked second in the world, right? So you have a lot of capacity, you have a lot of people with a lot of expertise that are here. So we have a very rich kind of research environment that's not only present in Riverside. We also have bee research in San Diego, a little bit in Irvine, and certainly a fair bit of that is happening in Davis. Right? So a very active and nice research environment. And then, of course, we have the Central Valley. Right? It's like California is the biggest producer of agricultural goods. In the US, it's something worth $8 billion a year. And the key example, I assume you have heard about that, is of course the almonds. At this point, about 85% of global almond production occurs in the Central Valley. They are fully dependent on bee pollination. That means that each year around this time now, about 1.2 million hives have to be transported there. It's the biggest man-made pollination madness in the world, right? And everyone knows about it. The US only has 2.8 million hives left, right? So a little bit less than half of the bees actually have to be transported to California to achieve this. So California really dependent on pollination, not just for almonds, but also for other parts. And then, like, I think where I kind of also get excited is the really kind of unique bee diversity beyond honeybees that the southern part of California has. I just show you these two pictures. It's just kind of breathtaking, right? So California has more than 1,500 different bee species. I think only even just in Yosemite, you have more than 400 bee species. This is a global red hot spot for bees and bee diversity. So it's absolute dream to see them here and also to study them here. And finally, you have what we call survivor bees. And these are bees that basically, we had an incident in Brazil in 1957 where researchers crossed two bee species, and that resulted in something that we refer to as the Africanized bee, and sometimes the killer bee, but I don't think that's actually an accurate name. So the Africanized bee was really successful, and it spread through the Americas. It also kind of entered the US, but the Africanized bee doesn't seem to be liking the cold that much. So it's present in the southern part of California, but it's not going any much further than Fresno. Right? Now, at the same time, from the north, we still bring in managed bees. Right? So what we have here around Los Angeles is like a hybrid zone. And that has just recently been shown is like the bees that we have here, the honeybees that we have here are extremely genetically diverse. These are neither pure Africanized honeybees, these are neither managed bees, this is a mixture of these things. And at the same time, you have some kind of continuous selection, because if some of these bees get too defensive or too aggressive, they are eradicated, they're taken out. So what we have now, and that's what we have started to collect, is like we have bees that are actually not that defensive at all, but they live without our intervention. They manage the environmental stressors that kills the managed bees. And I think we can climb up there into the trees, we can bring them down, and we can deploy the type of technology that I've shown you before, the type of approaches, and we can understand of how they do it, and we can use that to our advantage. Right? So I think like this southern part of Southern California is a bit like Noah's Ark of bees. It's an absolute dream to be here, right? So I don't regret a second to have moved here. And so with the beekeepers, like you need to have some platform, and that was the birth hour of the Center for Integrative Bee Research. We already had that in Australia. And integrative here means like we really want to kind of have this close relationship with beekeepers to work on tools, right? So the idea is here to use that knowledge and that science power to develop new tools that the beekeepers can deploy. There's no pressure on them to do so, but in our kind of philosophy is like, if we develop useful tools that actually work, the beekeepers will take that up, and they will be better able to kind of manage their bees. And the way this is organized is shown here. So we have these three kind of areas of research that I've shown you before, right? We have the molecular sciences. So these are all these kind of people that are interested in the James Bonds, in the agents. We have ecology and evolution, that's where I come from, like going out and studying bees in nature. And if you talk about tools, it's about engineering, right? So we bring these three kind of areas of research together. We deploy that in the honeybee, and we then create outcomes that broaden our understanding of bees, but at the same time, they help us to basically conserve honeybees, but in the long term also other types of bees. And here, like what we identified are three areas that are of specific interest, and that came back from the beekeepers. The first one is like the idea to breed better bees. So like we don't have sick bees, we don't have dying bees, we have bees that can actually cope with the problems around them. We need better ways to protect bees that have been exposed to stress. So we need to be better able to manage their health, but we also have to be able to better monitor them, especially under field conditions, right? So I cannot kind of go into every single kind of thing that we do here, but I give you some quick kind of ideas, if I still have a little bit of time, of the type of things that we do around this. So if you talk about bee breeding, we are going out and we collect these survivor bees, right? And we have one PhD student, which is called Kennedy Strong, and she is looking at those bees because they resist one of the most dreaded kind of parasites, and that is this varroa mite. And what she does is she puts these mites in a little arena. You can see this here. So the mites are still kind of locked in, and we have these larvae around here. And then she removes that kind of central part, and then the mites can actually choose their host, right? So they can run around here. You can see how the mites run around on the plate, right? And we then look at like whether the bees are able to kind of respond, whether they recognize that there's a mite around, and whether they respond to that. And what we found is, yes, indeed. Tolerant bees that cope with the mite naturally, they very well recognize that the mite is around, and they raise immune system. They raise agents that can take care of the mites. Right? We are not as far as with this Nosema spore, but we are on the way to basically replicate that. Now, if we can do this, of course, it would be really cool to get those bees together and set up a breeding program where we get bees that are manageable, so low defensiveness, but they are naturally kind of having the agents that they can resist to that mite. So we don't have to throw chemicals all the time onto these bees. And that also means we don't find these chemicals in the human food chain if we consume honey, right? So this is about the breeding program. I already talked about kind of, medication and the development of substances that we can use to cure that Nosema spore. There's another component I just briefly want to mention. And that is like, we have to produce food for an increasingly larger number of people. Right? And that puts enormous stress on agriculture. And so they will have to protect their crops, whether we like that or not. Right? So the idea that we could just get rid of insecticides is just not going to happen. Right? We started to understand how bad that actually is for the pollinators. And we did experiments like this here where we exposed bees, beehives, entire beehives. We fed them with pollen or with insecticide spiked pollen at very low concentrations. And we also kind of then collected bees and we infected them with that parasite that we have a cure against, that Nosema parasite. So we ended up with like 4 different groups of bees here, right? That either are completely happy, or they either have a parasite, or they're exposed to the pesticide, or they actually got both. And we looked at here, just one of the results here is like how strong is the immune response, right? And what you can see is that like the infected bees and the uninfected bees that are exposed to the pesticide, they already have a lower immune response. But if you combine this here, if a bee has exposure to pesticides, but then also gets exposed to the parasite, that really hits these bees, right? And so we call this a synergistic effect, right? So like these two things actually, they accelerate the damage on the bees. And that's really bad because we don't necessarily want to all the time treat the bees against diseases, right? We want them to actually mostly cope with them by themselves. So how do we get around this? Well, obviously we need pesticides. Obviously we don't want to treat the bees all the time. Our idea around this is, why not telling the bees not to go to certain plants if the pesticide is applied? And that resulted in a project that we called Smell to Repel. And this is driven by Boris Baer, which is my wife. So she's running that experiment together with a company actually that is really interested in that. And the idea is like, okay, we can monitor our crops, but if we have to use insecticides because there's an incursion, we want to deter the bees while that insecticide is there. And the way she's doing this is that like we started with some chemicals that we on the computer basically could identify as possibly being kind of repellents for bees, right? And she's then kind of using these little arenas here. So you can see 6 of them where you have bees and they are offered then honey because they've been starved, or honey that is spiked with a possible repellent. And you can see here H refers to honey, so they're really hungry, so poof, they go to the honey, but they ignore the repellent, right? And to our surprise, we don't only have one repellent by now, we have actually multiple of them. We have 6 of these repellents. And I think that kind of has great potential, right, to protect bees in the future, but it could have other appliances, right? We could use that like a mosquito repellent for people that are allergic. We could also use it in housing to avoid that bees actually move where we don't want to have them, let's say around schools or around kindergartens and things like that. All right, so that is like, you know, health management and managing stress exposure. What about monitoring of bees? Now, you know, if you have a dog or cat, you go to the vet, right? And the vet is specialized in looking after your animal, right? And there is ample of ways how the vet can intervene. It can vaccinate your dog. We have operations, we have medications. Now, we have the bee, and the bee is the 3rd most important animal for human mankind. The vets have never really kind of taken up to look after the bees. We have very little to almost nothing, certainly nothing compared to what is available for every other domesticated animal that we keep. Right? And I tried to kind of get them on board, and I even offered to give lectures to kind of, you know, I thought like, wow, there could be additional income for that, but they were not necessarily overly kind of interested in this. So we took a different approach. So we have happy hives normally, and we also know that, like, you know, there could be baddies coming along like these mites. Right? How cool would it be if we would have an electronic device in the hive that basically smells in there? Because we know that bees recognize that. We have seen that with our own research. Bees recognize the mites. Bees recognize when they are sick. Bees can develop a fever, right? So they communicate with each other. They communicate with each other when they're sick. They are communicating with each other when they're hungry. Can we smell in? It's not a listening in, it's a smelling in when the hive communicates with each other about problems. And if you read that bee language correctly, that sensor basically sends a signal on an iPhone of a beekeeper and says, not only there's a problem with your hive, but that's the problem. Right? So we call that an electronic veterinarian. We basically just replace the vet by an electronic device. And that might all sound like, you know, Star Wars or next century stuff, but here are our sensors, right? So for the last 5, 6, 7 years, we deploy a lot of sensors into hives. This is an experiment that we ran last autumn, where we now have like multiple sensors that we put in that measure different kind of things at the same time. And we then, like, you know, we stress the hives, we stress half of the hives, and we look at whether how sensor data that we get out of the hives actually can explain to us how healthy or unhealthy the hive is. And here's just one quick example, and I'm coming to the end of my talk, but you have here, like over a 10-week period, we have a very simple measurement, which is just temperature, right? And you can see here several curves. The red one is the temperature in the brood chamber, and bees regulate the temperature inside the hives very accurately, almost as accurate as we regulate our temperature in our own bodies, right? It is very closely, it's actually very close to our own temperature, right? They heat it up in winter, they cool it down in summer. So that's the red curve, right? And these two black lines are actually where the bees want to have it. That's the ideal kind of temperature inside the hive. Outside of these, the larvae start to suffer and to die. Right? And the blue line here is the outside temperature. Now, what we can see is that like the outside temperature is fluctuating over time here. The inside temperature is regulated, right? And that was a control hive. And then we have here this pink square here. And that is when we had predicted a really hot summer, and there are quite a lot of hot summer days in Riverside. So what we did then is like we had a second group of colonies and we put ice bags on top of them. And you can see that like the temperature in the box is actually kind of then decreased, right? So the environment around the beehive itself, we could actually decrease, but you can also see that the bee's ability here to regulate the hive temperature is much more accurate than it is up here, where they didn't receive any ice, right? So we help the bees here. And the interesting thing is here, even if we stop adding ice here at the end of this heat wave, those bees down here were still better able to continue to monitor their hive temperature. They had a follow-on effect. But in general, it's like the idea is like even something as simple as temperature, if you compare, on the box to where the brood is kept, can give us a lot of information of what's happening inside of the hive. So what we use now is we use machine learning to develop algorithms that basically can predict if we have a certain dynamics in a hive, like here, like whether that causes in the long term problems, or whether the bees will be able to basically monitor that by themselves. So we see that like this idea to deploy sensors works in principle, even with very simple measures such as humidity or temperature. And after those 10 weeks, there was actually difference. So the bees that we provided with ice had more honey than the ones that were not provided with ice. So these things actually impact how well the colony performs. All right, so the very last thing I want to mention is some project that we started there. I started with bumblebees, and I did some larger segment on honeybees. We also start to go back to native bees, and we started a project now to look at bee hotels. And I mentioned that to some of you at the very start. We also want to know like a little bit more about the other bees that are around here. And one way we propagate, or one way we try to kind of encourage people to care about bees is to recommend bee hotels, because it's an easy way, a much easier way than if you keep honeybee hives. But no one really knows how effective they are, or whether any threats to it, or what actually lives in there. So we deployed bee hotels. We monitor them now for 2 or 3 years. It's a citizen science project where the students on the campus are involved. We call it Airbnb, and where we try to actually determine, what is living in there, what lives over time in there, and like, are there any risks? Can we basically modify the setup to attract more, or to, for example, attract more rare species? So that is something that basically brings back the technology and the approaches that we develop, not only to honeybees, but also in a larger context to these tremendous kind of amounts of native bees and other insects that live here. And I think that's it. I hope I didn't go too much over time. If you are interested in keeping up what we are cooking up all the time, we have that website, that CIBER website. And we also have a Twitter feed and a Facebook page where if we publish a new paper, or if we have an outreach event, or if we have a seminar or a conference, we announce that on these two on that website. Yeah.
Okay, thank you very much. Boris, I'm very glad I invited you. That was fascinating. At least I thought it was fascinating. I didn't realize that UC Riverside was that advanced in the world of entomological research. I knew they had a lot of research. I didn't realize we were that well thought of. So I guess I will kind of lean a little more heavily and encourage a little more interaction with UC Riverside. I have a question about the bee hotels. Do you know if there is any cross-infection occurring in the bee hotels? In other words, are you encouraging, is it possible you're encouraging cross-infection between species or amongst single species of diseases and parasites with the bee hotels?
We don't know. It's like the issue here is that this is a crossover to the commercial world, and then we have a lot of opinions. I think there's very little research done around that, and that is one of the issues. Now, look, generally, bee business, pollination, so it's just really dirty business. Okay, bees. It looks so nice when a bee visits a flower, but these flowers are loaded with different diseases. We have transfer of diseases among bees. We have transfer of diseases between bee species. It's not necessarily something that is unnatural, right? Maybe like our human activities really kind of curb that up and make it worse, especially when we keep unhealthy bees. But in principle, bees are used to diseases, and as we show, and that's the same for bumblebees and for other bees species as well, they can defend themselves. So it's not so easy to actually dissect that out, right? If you can look at like bumblebees, you can look at these bee hotels, you can detect the parasites. Yes, they're there. Are they still alive? I don't know. Are they propagating in other bee species? We don't know that either, right? So for me at least, there's a very open field of questions that we haven't asked, but that we should ask, right? As I said, for me, seeing a bee that is parasitized is not yet ringing all the alarm bells. It rings alarm bells when you see that these parasites propagate and start to really hurt the bees, and they can't deal with these parasites by themselves. So we don't know, and that's the entire idea behind this project, right? That like if we deploy on campus 30, 40 of these bee hotels, we can go back on a regular basis, we can take samples, we can look at the larvae and at the offspring, whether we see propagation of these diseases, whether it impacts the populations in the long term and things like that. And I think that's what needs to be done. It's also what I feel really important is that all of these things will tremendously differ where you are. So, you know, if somebody in Europe or somebody in Southern Australia sees a parasite in a bee hotel, there will be very difficult to kind of transfer that to California or to Panama or wherever you are, right? So I think like it needs to be looked at in a very kind of local context with local species of bees and communities around.
Okay. Have you seen any difference in effectiveness of these bee hotels? Have you seen one brand or a style that is more or less effective?
Yes, yeah. There are some bee hotels out there that are just nonsense. Like, you know, and I collect them. I don't want to be too trashy on the one side about this. I don't think people do that deliberately, right? And I also don't want to kind of necessarily kind of say, this is the bee hotel that you all have to buy, right? Because in all honesty, I don't know that yet. What we do is we sample the variety. We have these more general kind of bee hotels that if you go to Amazon, there is a huge variety of these things around. You can look that up by yourself. Most of them to me look like the basic design is always the same. You have these bamboo thingies in there, and then you have some holes in some wood, right? And the rest changed around because it's based on the aesthetic needs of the customer. So these things are built so we buy them. No one asks whether the bees actually like it, right? And that's fine with me. But I think like if we could come up with better guidance for people how to deploy them, how to maintain them, and also maybe better ways how we could build them in the future, they could just improve like the effectiveness of this thing. So similar to like the other things that we do, we try to develop tools that help us to actually help the bee populations out there. And I think we are at the start of that. As I said, we have some bee hotels around. I got donations of bee hotels that we have. And COVID did not really help to get something like this up and running because we were all sitting at home waiting that we could actually leave the house. So I really want to push this now over the next 2 or 3 months. We have like the team together, and there are, at least in the beginning, we have about 10 to 15 PhD students in the department that will be involved in this. The student organizations at UCR will be involved in this. So we have more than enough interest, but we also have the opportunity now to actually meet and go out and do stuff again. We are allowed on the campus again. Yeah.
Have you seen any of these bee hotels that might be more effective? Have you seen one that you like yet?
Yes, so I like the ones that are more diverse because it attracts more critters, right? So if I look at my bee hotels, as I said, maybe I have to start, we might have to rename them insect hotels, but some of them have attracted moths. Some of them have attracted monarch butterflies. So they pupate underneath the bee hotel. It was like, woof, okay. Some of them attract a lot of different wasps. I had like, so I have a huge variety of different kind of creatures and insects that live in there. And you have these simple ones that normally kind of, they have just the holes or they have just the bamboo. Generally, I see less biodiversity around those than those that have like different structures and are more complicated, right? But I want to quantify that in a scientific way, right? So these are observations, and we use these observations at the moment to basically set up a larger scale experiment around this. And that we haven't yet decided, like, do we kind of have certain areas on the campus where we deploy multiple hotels and we see how they do comparatively, or do we just deploy them over a large area and we want to know, like, is it the environment? Is it like where they hang? How much sun, how much shade? We haven't decided on the exact deployment on this. This will all be guided when we will have a first overview of the bee hotels that we already have. That will generate the questions, and that will generate like the setup that we will use to really kind of roll that out on a larger scale. Okay. Does that make sense?
Yes, it does.
Yeah.
Any other questions?
Hello, yes. I wanted to ask, the superlative really caught me about how many parasites that honeybees have. And besides the size of the hives and the complexity of them, the density of the bees, is there any other leading explanation for why that might be?
Yes, yeah. I think in honeybees, it's very clear that mankind has domesticated bees tens of thousands of years ago. So we see the beekeepers depicted in old Egypt, even in China, even further back one. And we traveled around with them. Wherever we went, we took the bee. And that meant that the bee came in contact with diseases that, if you talk about the European honeybee, the managed honeybee, that were not original diseases. So the varroa mite is not the original disease of the European honeybee, but it comes from another bee species. Nosema serrane, this fungal disease that we work with here, is not an original honeybee disease. The hive beetle is not an original honeybee disease, and so on, right? So they collected, as we traveled around, they collected additional diseases, and we started also to breed bees, right? We don't want to have defensive bees. We want to have nice bees, and we want to have bees that produce a lot of honey. So we lost a lot of genetic diversity. The genetic diversity of the US bees is extremely deprived. That's not like, I showed you in the beginning how important genetic diversity is. That was the start of all my work, right? So I think that favored that bees have diseases, that they acquired new diseases, and also that they lost the ability to really kind of throw them out or suppress them efficiently. And I think at that step where the beekeepers are fully on board, I think that the beekeeping needs to go beyond of, we need a lot of honey, we need a lot of bees, we need like maximum kind of economy. Bee health needs to be almost at the first step there, right? And that brings, like, with breeding of more genetics, with helping beekeepers when they get sick, with monitoring of bee health, I think that all kind of guides us towards these new tools to do this. And my hope would be that if we keep healthier bees, that will also have, like, that will certainly reduce the risk that we do any damage to native bees, which would be absolutely devastating, because we know that the native bees are not only playing an important role, and I dearly love them, but it's like we know that they suffer much more from these stressors than the honeybees, right? The bumblebees, the native bees, they really suffer from pesticides. They really suffer much more from environmental change and global change, because they can't go away, right? We feed our bees, we help them, we move them somewhere else. A bumblebee colony can't do that.
Okay. Any other questions? Well, you know, I think that UC Riverside is welcome to take advantage of Lorquin. I'm perfectly happy to use Lorquin as much as I can to distribute or encourage interaction and information from UC Riverside, because that's what we are as an entomological organization. And I would like to continue to interact with UC Riverside. It was one of my first choices for a PhD myself. So I would like to continue it in the future, and we'll probably will see more speakers from UC Riverside talking here. If there is any other questions from the audience. Boris, that was a wonderful talk. I loved it personally. I was fascinated the whole time. I hope we can maybe see you in the future. Your research was very interesting, and you presented it in a very digestible manner. So we greatly appreciate it, and we hope to see you again sometime in the future. You're welcome to join us at any time. I will try and keep you on the list of announcements for Lorquin.
Yeah, well, the department has a lot of entomologists, right? So I'm sure there are others that can give updates, whether it's on bees or other stuff. Mm-hmm. But otherwise, like, you know, I gave you a very crude overview over everything, but like there are lots of people behind. I tried to show some of the faces. Maybe I should have added a picture of all the people that actually contributed to that. So if you want to have more detailed information, I don't know, about the medication, about the bee hotel, so it's like there will be people around that maybe kind of can give you more details and more excitement about these into these individual projects and activities that we run.
Okay. Well, thank you very much.
No worries at all.
Yes, thank you very much.